for exam 2, so you'll have a full week before we do the exam. If we have chapter 11 done by Monday, plus some time to answer questions on the exam, then we're going to be right on track with the syllabus. Okay? Any other general questions? Okay. So what we're going to do is we're going to continue with chapter 9 today, which is on the bacterial genetics. So before we get into that, let's do the 2 questions that we usually do. So from the previous lecture, here's a question about plasmids. Let's get. Anybody lose their contact lenses? Yeah, it looks like 1 minute should be enough. Okay, 3, 2, and 1. So what's the answer? Here we always got to do this for whatever reason. Okay. So this is a very important point, and we'll come back to this a little bit later, is these plasmids do not carry genes that are essential for the survival of the bacteria. In fact, it's very common for the bacterial cells to lose these plasmids if there isn't some selective advantage. But we'll talk about that a little bit later. And then for today's lecture, yeah, that one's pretty easy. We'll do the 1 minute on this one, make it easy. How do I know? How can I tell? In the end, it's all about knowledge. 3, 2, 1. All right. So this one's obviously what? These are the viruses. Okay. So all right. So this is the one. Yes. Okay. That's actually what we're going to be doing here. But the episomes are. So the question is, what's the difference between plasmids and episomes? Well, episomes are really a subset of plasmids. Some plasmids, as we're going to talk about here, the F factor, just to review for where we were before, are episomes in that the plasmid can either maintain itself independent. So here's the main chromosome. This is not drawn to scale. So the plasmid can be independent, replicates independently, or it can insert itself and become part of the main chromosome. Episomes are plasmids that can integrate into the chromosome. They become part of the main chromosome, and they can come back out again and be autonomously replicating molecules. So this F factor is a type of episome. Yes?
Bacteria have a DNA that's one molecule, but those molecules are still double-stranded?
Yes.
Okay.
Yes. In all living cells, except for viruses, all living except viruses aren't living cells. So in all living cells, to my knowledge, the genomic material is always double-stranded DNA. It may be linear, it may be double-stranded, it may be segmented, but it's always double-stranded DNA. Okay, so what we're doing is we're talking about the sex life of E. coli here, or more scientifically, how does genetic information get transferred from one bacterial cell to another? So this is with E. coli. We're going to use that as our one example of this process of conjugation. So there's a physical transfer of DNA from one cell to the other. Now, this is a review of what we talked at before, is this process of conjugation. The donor cell has to be an F plus cell. F plus just simply means this E. coli cell has a copy of the F factor, which is a plasmid in this form. It is an episome. We'll talk about that in just a minute. And all that's going to happen is a copy of this F factor is going to get transferred to this recipient cell, which is designated as F minus. So F minus simply means the cell doesn't have the F factor. The only difference between F plus and F minus is the presence or the absence of this F factor. So you have the pilus, which is this protein conduit, which actually allows a physical transfer of DNA from one cell to the other. And the key point here, and I think it makes it more in this next slide, is right here that only one strand of the double-stranded DNA is being transferred. What would happen to the donor cell if both strands got transferred? It would lose the F factor. So the donor cell is going to remain genetically the same. A single strand of DNA from that F factor is being transferred to the F minus cell, and then the DNA repair systems can convert the single-stranded DNA into double-stranded DNA, and now you have two F plus cells. So in sort of the classic designation, the F plus was considered the male cell, F minus was the female cell. So this is actually a sex change operation for a bacterium. Okay. Now this process is very efficient because this F plasmid is very small. It probably doesn't take more than a minute or two. So the change from an F minus to an F plus cell probably approaches nearly 100%. All right. Now, before we talked about this, we talked about these experiments that Letterberg and Tatum did, showing this transformation where there was genetic exchange of information between these two different E. coli cells, but there were genes involved in metabolism, making amino acids and such. Would those genes be on a plasmid? No, they had to be talking about a transfer of chromosomal genes. What they were looking at were HFR cells. So this F factor is an episome. It can integrate itself and become part of the main chromosome. So that cell no longer has a self-replicating plasmid molecule, but all those genes that are on that F factor are now present in the chromosome. It got its name for high frequency of recombination, and it happens about maybe 1 out of every 10,000 cells. Since that F factor is in the chromosome, it can still undergo conjugation just as it did when it was an F plus cell. Now here's the important point. The F factor can integrate at random into the chromosome. But the transfer of DNA from the HFR cell to an F minus cell always begins at the point of insertion of the F factor. So the F factor can go in in some random location on the chromosome, but transfer always starts at the F factor. Okay. But since it's part of the chromosome, you can now transfer not only the genes on the F factor, but some of the chromosomal genes. How many of those genes get transferred depends on how long conjugation is maintained. That pilus is a very fragile structure. Once that structure breaks, then conjugation is over. That can either be accidentally or done on purpose, as in these mapping experiments that we're going to talk about. Which genes get transferred depends on where that F factor inserted. And we can use this as a mechanism for mapping the genes around the E. coli chromosome. Now, transferring the entire chromosome, in contrast to that F factor, takes about 100 minutes. So it takes over an hour and a half to transfer the entire chromosome. Do you think it probably is very efficient? Does this usually happen? No. But the whole map, the genetic map, is put together instead of map units like we did when we talked about the Drosophila genes. That was based on recombination percentages. Here, it's going to be based on the time of transfer, the time it takes to transfer one gene to the next. And so the whole map is based on this kind of clock of 100 minutes. So the distance between the genes is measured in minutes. Okay. So here's this rather complicated slide. All it's really showing is we're really going to look at how long it takes for each of these genetic markers to show up in the recipient cell. So we set up this conjugation experiment. We just mix the two strains together. And what the graph is showing you is what? Which gene transferred first? Which one? Okay. So the azide resistance transferred first, then the TON resistance, then LAC plus, and then GAL plus. So what this is really showing you is the first gene after 2 minutes, azide resistance transferred, and then 2 minutes later, TON resistance. 6 minutes later, LAC plus, and 9 minutes later, galactose. So you're just measuring the time that it takes for each of these genes to transfer. Now, if you just had this one strain, could you put this together as an unambiguous map? Probably not. It's going to be very difficult to get the entire chromosome keeping this together for 100 minutes. Well, what we do is we take advantage of the fact that this F factor can integrate at random into the chromosome. So, a couple of things before we show you that just to keep in mind. So these different HFR strains are going to have the F factor integrated at different places. What we mean here by directional gene transfer is that F factor can insert itself in two different orientations. If you know something about the polarity in the 3' and 5' ends of DNA, you understand there's a directionality to it. For now, let's just say the integration can go in either of two directions. It's random. And in one orientation, transfer will go clockwise as we draw the map. Clockwise as we draw the map. And in the other orientation, it goes counterclockwise. So here's HFR1. This is just one of the strains. It has the F factor inserted over here between the gene for azide resistance and this gene involved in leucine metabolism. And this arrow indicates that transfer is going to go in a clockwise direction. So you're going to transfer leucine, threonine, thymine, histidine, galactose, lactose, proline, azide resistance. That's the order of the transfer. Here's a different one, just called HFR5. Now the F factor, instead of being over here, is located over here, and it's oriented in the other direction, so the transfer is going to go counterclockwise. And so the first gene to be transferred is what? Thymine and threonine, leucine, azide. Right? So you get a whole bunch of these. And you got 6 different HFR strains. And can you see from that how you put the map together? Really? Oh, good, good. In case you don't, what you're really doing is all you have to do is reorient them, get them all going in the same orientation, depending on which way it inserted, and you just simply see that the genes all line up in the same order. So once you do that, it doesn't matter which of these strains you use, the maps are in the same order and the distances between the genes are the same. And this is how the genetic map of E. coli was put together originally. Of course, now it's been sequenced about 100 times. Okay? See how that works? All right. So let's try this. Let's see if. Okay. Remember, you can collaborate on these questions. No, answers aren't coming in quite as fast, though. All right, we'll go to 1 minute and 30 seconds on this one. 3, 2, 1. Okay, so what's the answer? All right. So here's the transfer. Okay. So do you see why would Tane and lac be most likely transferred in most experiments? Transferred together? Yeah. It's only 10 minutes apart. But what about lac and azide resistance? About how long is it going to take before the azide resistance transfers? Nearly 100. All right. The transfer is starting here at the tip of this arrow and going clockwise. And so this is going to be one of the last sequences to be transferred. Okay. But otherwise, that's all there is to it. It's just you're just mapping them together as they transfer out. Yes.
Do you go in the direction of the arrowhead or away from it?
The way they're doing this, and it doesn't really matter. You get the same result. They're trying to use the arrowhead to show this orientation. So if the F factor, let's say in another strain, inserted in exactly the same place but flipped in the orientation, then azide would transfer first, and the lac would be the last gene to transfer.
Okay.
Yes.
The red arrow is going clockwise.
Well, it's showing here that this tip of this arrow is going to go first and pull everything behind it. So the transfer, the way they've drawn this.
The way I see it is that the arrows break it and continue so black and
Yes. So in this, the way they've drawn it, lac would go first, then ton. You go all the way around the circle, and azide resistance would be last. Okay. I don't. Okay. Let's try it in another form. So let's try this question. This would be more like a question that I'd probably have on the exam. I'll go to 1.5 minutes on this one.
yeah.
Okay. So what's the answer? All right. So they all start with C. Okay. So if you look here, let's just take C. G is on one side, F is on the other. So you can immediately eliminate any that don't have one of those two letters. So for instance, C is definitely out. Well, what you're looking at is it's just simply in an order. So let's see if we start. We got C, F, D. And right here we have F and D. So we could have C, F, D, E, B, A. That would be one way. We went the other way, didn't we? So we got C, G, A, and then we come back up to A, then B, E, B, F. Okay, that's all there is to it. Any questions? Okay. A little bit about these plasmids. Probably the two most common genes, or elements, if you want to call them that, you're going to find on plasmids are genes that encode drug resistance, antibiotic resistance, and transposable elements. We'll leave transposable elements for another time. In terms of MRSA and these other drug resistance infections and such, this is the main culprit. These antibiotic resistance genes are on these plasmids. You're getting a natural selection, especially in a hospital environment. The bacteria get exposed to the antibiotic. It selects for that very small population that has the drug resistance, and then that population explodes because the bacteria reproduce so rapidly. Also, this project of conjugation doesn't always follow species barriers. I think there's a slide coming up about horizontal gene transfer. So these drug resistance markers get transferred between other bacterial species very efficiently. Let's see. What was I going to say? All right. Don't know if this is in the book or not, but just a question. So several years ago, they found the remains of some explorers up in the Alps that had perished in an expedition in the late 1800s, and the bodies have been frozen up there for all these years, and they found them 10, 15 years ago, something like that. In doing autopsies and examination of the bodies, they found bacteria in these specimens that were resistant to antibiotics that had not yet been invented at the time. Now, assuming that it wasn't due to contamination, which it could have been, how would you have bacteria present that are resistant to an antibiotic that had not yet been invented? Seem possible? Yes.
The antibiotic was created to attack the
Right, but these bacteria presumably were pre-existing, already resistant, because they're in the bodies of these, frozen in these bodies, these individuals who were there before these antibiotics were invented, decades before. Yes?
Was there another antibiotic that they had resistance to and then they mutated?
Somewhat similar to that. What you find is a lot of these antibiotics that we have are, like penicillin and such, are derivatives of naturally occurring compounds. So most likely these bacteria were already resistant to this naturally occurring compound. We subsequently discovered it. I don't know if they still do it. It used to be pretty common with a lot of pharmaceutical companies. If you were sent to a conference in another country or even another location in the U.S., you had to bring a little bag of dirt back, and then that was used to try to isolate for bacteria to try to find a mold and such that would produce new, unique antibiotics. Okay. All right. So at the beginning of this chapter, I said there were three ways we were going to talk about how genetic information, basically DNA, physically gets transferred between bacteria. Conjugation is the main one. That's what we already talked about. The next two I'm just going to talk about very, very briefly because they're really not used for genetic mapping any longer, but they do have, one of them at least has an application. That is transformation of bacteria. At the beginning of the next chapter, I'll talk about a landmark experiment done back in 1929 that where Griffith discovered this phenomenon of transformation. What we mean by it is the ability of the cells to take up DNA from their environment, essentially get that DNA across the cell wall, across the cell membrane, and incorporate it through recombination into their genome. Some bacteria are naturally competent. That is, what we mean by competent is they naturally have the ability to take up these DNA sequences. The Streptococcus that Griffith was working with, naturally competent. Our old friend Neisseria gonorrhea is naturally competent. Although interestingly, it can distinguish its own DNA sequence from other DNA sequences. Most bacteria aren't. And so the one that we use in the lab most of the time is E. coli, which is here. We like to work with E. coli. We know a lot about it. We have a lot of strains. The lab strains aren't pathogenic and such, but we have to play tricks on it because it's not naturally competent to take up DNA. So one of the things that we'll show you how to do in the, I think we might do this in the micro lab, we definitely do this in the biotech lab, is when we want to genetically manipulate these E. coli and put DNA and plasmid sequences and such into them, we have to make them competent to take up that DNA. A very inexpensive and reasonably efficient method is simply soaking the cells in calcium chloride. And in a way in which I don't think the biophysics is still completely understood, this high concentration of calcium does something to the cell wall and the cell membrane, and that, along with a little heat shock at an elevated temperature, facilitates getting the DNA into the cells. What's much more efficient and much more widely used now is a process called electroporation, where we literally electrocute the cells. So we have the DNA, the cells are suspended with the DNA, and then they get a very brief pulse of a very high voltage. With bacteria, probably somewhere in the range of about 2,000 to 2,500 volts, but just for a few milliseconds. What this very high potential does is thins out the membrane and causes little transient pores to form in the membrane, and the molecules get into the cells at that point. One advantage of it is it works with just about any cell type, eukaryotic, prokaryotic. You have to play with the electrophoresis, the electroporation conditions, the time, the voltages, but it works with just about any cell type. The only downside is you got to have a few $1,000s to be able to spend on it to buy the equipment. But again, we'll show you how to do both of these in the biotech lab. All right. Early on being probably in the mid-1900s, transformation was used as a way to do fine structure mapping of genes in E. coli. This conjugation is still rather crude. We're not going to go into this. I just want you to be aware of it because it's now pretty much irrelevant because of genomic sequencing. And so just a few little ideas on this. What's happened now is we've reached a point with the DNA sequencing. It is so fast and so cheap and so efficient that these other methods for mapping genes have pretty much are obsolete at this point. The genomes in bacteria are pretty small relative to our genome. They're in the range of 1 to 4 million base pairs. On a comparison scale, our genome is about 1,000 times larger than this. So this slide isn't in yours. I just put it in today just to show you how fast this went. Basically, and for most of you, in your lifetime, in your general lifetime. So way back in 1995, when you were wandering around in Pampers, and some of us were teaching this course, the first two bacterial species completely sequenced their genomes. They were published in 1995. Anyone guess what the first bacterial sequence was? E. coli? No, it turned out to be Haemophilus influenza, which causes what? No, the bacterium Haemophilus is a misnomer because for many years it was thought that it causes the flu before it was known to be caused by a virus, but the name is hung on. So anyway, so 20 years ago there were 2 bacterial genomes sequenced. At this time there's more than 30,000 bacterial genomes in the NCBI GenBank DNA database. The GenBank DNA database is publicly accessible. You don't have to have a license or anything. This is your tax dollar at work if you pay taxes. Okay. We're now up to what's really being called third generation sequencing. So I used to go through the chemistry of DNA sequencing, which now is pretty much irrelevant. Plus, you wouldn't do this by hand. This is all something you outsource at this point. And what I wanted to show you is that it's gotten so fast now that most of these facilities could sequence an entire, if you found some new novel bacterium, they could sequence the genome in a matter of hours. You could almost have it done before lunch. And at this time, there's at least sequence data for at least 50 different bacterial phyla in the GenBank DNA database. This is that slide on the horizontal gene transfer. What we mean by horizontal gene transfer is a transfer from species to species that's not going through replication. So it's staying at the same generation, and it's really a transfer between different species. Because we have so much this genomic data now, it's becoming clear that a lot of these bacterial genomes are composites of genes that have been pulled from other species, estimating that almost 1/5 of the genes in E. coli were actually probably acquired from other species. So the bacteria do this. They don't seem to recognize species barriers all the time, and they seem to be able to transfer this genetic information very efficiently. So one of the things, though, that's been coming from the sequencing, haven't heard about it for a while, but every once in a while, I can never remember the strain name. There's that pathogenic strain of E. coli that ends up in hamburger and shows up in the local fast food restaurants and such. So it's a pathogenic form of E. coli. Traditional genetics would have taken who knows how long to try to figure out, okay, what genes are present in this pathogenic strain that aren't in this non-pathogenic strain. Now you just sequence the genomes, compare them, and you find out what genes are different. Okay. The other thing that's changing is something called metagenomics. We've known for a long time that we can only culture a tiny percent of all the microorganisms in our environment. Up until now, if you couldn't culture it, you couldn't study it. You couldn't sequence it. But because of the newer techniques, you can get enough DNA from these very tiny environmental samples that you don't actually have to culture the bacterium to be able to get genetic information on it. Here's one example. It may have come from the textbook, or maybe I can't remember where I got it. But you basically, first of all, the microorganisms of the ocean have just been tapped. I mean, we're just barely getting started. This would include the eukaryotes, the algae, along with all the microorganisms and such. In this sample, they basically just filtered 1,500 liters of seawater, extracted DNA from all the microorganisms they collected, and just started sequencing it and identified 1.2 million new genes distributed among more than 1,800 different bacterial species. So this is really coming into what's now being called metagenomics, which is a genetic analysis of very complex populations. It's really been focused in on microbial populations. Maybe you take a sample from skin and ask how many different bacterial species are there. We only can culture a tiny percentage of them, but we just extract the DNA. And in this process of next-gen sequencing, it's beyond the scope of this course. But the take-home message is we can look at the genetics of very complex populations of organisms. This is expanding out all the way up into humans now. We have the 1,000 and 2,000 genome project. So what we mean by metagenomics is when we're doing a genetic analysis from a very large mixed population of individuals, and from that being able to tease out some new genetic information. Yes?
Do they find, when they filter out the water like that, and even when they don't, how do you know when you have a new gene, when you have so big letters on a computer screen?
Yeah. So the basic premise behind a lot of this is you start looking for a certain target gene. So you take something like a 5S ribosomal RNA. A lot of work has been done on that. And we'll get into this later on, but let's say you're doing this with algae or some eukaryotic organisms. These 3 ribosomal RNA genes are organized the same way in all eukaryotes that have been looked at. And there's a spacing region between those 2 genes. And since that doesn't actually code for a functional RNA, it's changed over time. And so you're really looking for how many different versions of that 5S ribosomal RNA gene are there. And from that, they probably concluded there's about 1,800 different versions. But in the meantime, they're using other sets of primers and such that are giving information. So it's a very, very complex. You can't necessarily say, oh, here's the new species, but rather there's about 150 different species here, and we've been able to identify some new genes and that sort of thing. So it's a lot of bioinformatics, but it's really being able to have the sensitivity to be able to sequence DNA from such a tiny sample of DNA, having the computer power to crunch all of those numbers, and also these massive databases. These are in terabytes of, multiple terabytes of data coming from these. Anything else? Okay. So the point I wanted to make here is, so we know all of this information about E. coli. It's the most well-studied, clearly the most well-studied prokaryotic organism on the planet. It's been studied genetically since the mid-1940s, 1950s. We know exactly how many base pairs of DNA, at least in a prototypical genome. There's 4,300 genes, and with all of this work, we still don't know what half of those genes do. And in many cases, what's coming out of these kind of studies is looking at the DNA sequences, begin to predict what those genes might be, and then you can do a reverse genetics type approach. What I really want to make from this point is even an organism as well studied and as simple as E. coli, half of those genes we still don't know what they do. Okay. All right. The last part of this chapter is just to touch a little bit on viruses. So we'll spend a little bit of time on bacteriophage, which are the viruses that infect bacteria. I'll give you the example of Lambda phage. It's a very, very well-studied double-stranded DNA bacteriophage. And then just talk very briefly about a couple of the RNA viruses, really in the context of being eukaryotic viruses. So as far as we can tell, all organisms are susceptible to some type of virus. Somewhere, I'm trying to remember, it probably goes back maybe 15, 20, boy, time goes by so fast. There was a book in the past where people had the idea that humans were the top of the food chain, but the thesis of this book was no, viruses are the top of the food chain. Okay. At its most fundamental basis, a virus is just going to be a piece of nucleic acid wrapped up in a protein coat. And it might have a layer of lipid around it for the animal viruses. By itself, it's functionally inert. It's just a piece of nucleic acid. It's just a chemical at that point. But viruses really, as you know, they're cellular parasites. So what the viral genome is going to encode are the genes that it needs especially for its purpose of being able to infect and replicate the cell. Then it's going to take over the host cell machinery and use the host cell to have its DNA polymerases and the transcription and translation. So viral genomes tend to be very small, whittled down to just the genes that are essential and unique to the virus. In other words, those genes are not going to be found in the host cell. As we've talked about before, viruses can be very flexible in terms of their genetic material. Some are DNA, some are RNA, double-stranded, single-stranded, circular, linear, partitioned up into multiple segments. Reovirus has 9 different RNA viruses, almost like little chromosomes. But no viruses have been found that have both DNA and RNA, which is what we find in normal cells. So if junior scientists were to find some new cell or something like that, or whatever you might call it, and if it only had RNA or DNA, it most likely is going to be a virus. Okay, so we'll talk about bacteriophages. We're really going to talk about Lambda phage as an example because it covers all the points that we want to make about bacteriophages, and it's an extremely well-studied model. Okay. So viruses, like people, have alternate lifestyles. Viruses can either be lytic or they can be lysogens. The simplest one are lytic infections. That means the virus is going, the prophage is going to infect the cell, it's going to replicate, it's going to make new viral particles, it's going to kill the host cell, and those viral particles are going to go out and terrorize the neighborhood and start infecting surrounding cells. A virulent phage is a phage that can only go through this lytic life cycle. It doesn't have this choice. Okay? Temperate phages, in contrast, have a choice. They can be lysogenic or they can be lytic. The lysogenic is very much like those episomes. Instead of after infecting the cell, instead of making new viral particles, it just inserts its genome into the host genome and, very importantly, shuts down expression of all the viral genes. So there's no viral proteins being made. The host cell doesn't even know the virus is there, but the virus is doing what it wants to do, perpetuates itself, because every time the cell replicates, the viral genome gets replicated. All right. Now, under certain conditions, when the neighborhood goes to hell, this lysogen can come out of the chromosome and initiate a lytic infection. So here, I'll show you how that works. Okay, so any questions? All right, let's break it down a little easier. We'll do the lysogenic part first. So here's the bacteriophage Lambda. Many of these, they all work the same way. They have this really rather complex protein coat that kind of looks like a lunar landing module. And the whole purpose of this is the DNA that's up here in the capsule head of the phage is going to get injected into the cell. So the DNA enters the cell. And in this case, so here's the DNA, enters the cell. If it forms a lysogen, it actually integrates and becomes part of the main chromosome. As I said, not only is it now part of the chromosome, so it's going to get replicated every time the cell replicates, but also none of these viral, these infectious genes are being expressed. So the cells divide, and they can just keep going around through this cycle. The cells keep dividing. It's almost like a time bomb, though. The lysogen is there, but it's not causing any damage to the cell. It's a little bit of a symbiotic relationship because these lysogens, these E. coli that have a copy of the Lambda genome inserted into their chromosome, cannot be infected by another virus of the same strain. So there's a little bit of a symbiosis going on. Yes?
So if the lysogen integrates itself into the chromosome, does it change the DNA of the host cell?
No, it actually just, there's in contrast to the F factor, the bacteriophage always inserts as a place called ACT for the attachment site, but it inserts itself without damaging any of the genomic DNA. It's not exactly the way it works, but it's literally just, so you can imagine just snipping the circle and just putting the DNA in there and then joining it back together again. So as far as E. coli knows, it's just this extra little piece of DNA. It's about 50,000 base pairs, so it's very, very small compared to the 4 million base pair genome. And it just continues like this. But under certain conditions, we'll talk about this later on when we do DNA repair. For instance, if this DNA gets damaged, it gets exposed to ultraviolet light, Then the phage can actually come out of the genome and go into the lytic cycle. So it comes out, it starts replicating using the host cellular machinery, it makes more copies of the DNA, it makes the proteins, starts assembling them into phage particles, and at the very end, most of the time there's about 200 of these phage particles. The last thing it does is makes an enzyme called a lysozyme, which degrades the cell wall and the cell membrane, and the cell just ruptures, and these viruses go out and infect new cells. So this is this temperate phage, because it can either be this lysogen or initiate the lytic infection. You can see this in the lab. This kind of cloudy field that you see on the plate is just a lawn of E. coli. Then each of these clear spots on here is where there was one E. coli undergoing a lytic infection, and then the viral particles came out, infected the cells around it, So if you actually took a toothpick and touched it to the center of that clear zone right there, that plaque, you'd probably pick up about 1 million phage particles. Okay. Any questions? Okay. So that's a pretty prototypical bacteriophage. This third method for doing genetic mapping was called transduction. Again, we're not going to go into how it was used. I put it here because what transduction means, DNA is being transferred from one E. coli or one bacterial cell to another using the phage. So what would happen is sometimes when the bacteriophage is packaging up its genome, I forgot to mention one of the first things that happens with a lytic infection is nucleases are produced that start breaking up the host genome. So that host cell is going to die no matter what. So there's pieces of genomic DNA from the host cell floating around, and once in a while the bacteriophage makes a mistake and packages up a piece of that host DNA, and it gets transferred to another cell. So just all I want you to do is know that this transduction is a process of transferring genetic information from one bacterium to another using a bacteriophage as the vector. And that it was once used for some fine structured genetic mapping, but I doubt if anybody's using it any longer. I don't think you fully appreciate the fact that I'm not doing phage mapping with you.
We appreciate it.
You appreciate it. It's pretty tedious. Okay, so while you think about some questions, why don't we try this question? We'll go to about 1 minute and 15 seconds on this one. Okay, what's the answer? D. There we go. So those are all mixed up. None of them are. So conjugation would be which answer? Would be B. Transformation would be C. And transduction would be A. Okay. All right. The last part of this chapter is a little bit on RNA viruses. Now, we're getting away from bacteria. We're getting back into eukaryotes. So these examples are primarily animal viruses. So we're going to look at RNA viruses. So I'll give you a couple of examples of viruses where RNA is the genetic material instead of DNA. Okay, you got to stay with me because this is going to get confusing. So these RNA viruses, we're going to talk about, let's see, positive strand, negative strand, and reverse, and the retroviruses. The positive strand and the negative strand. All right. How do I do this? All right. So there's an example there. So polio is a positive strand, rabies is a negative strand. Here's the important point. First of all, the genome is a single-stranded RNA molecule. Okay. So the virus is going to infect the cell. This is into an animal cell. And it needs to make more copies of its RNA genome. Right? So is this host cell, this is going to be a eukaryotic cell. Will this host cell probably have enzymes that can make an RNA molecule from an RNA template? Is that normal in cells? What cells do you know that do this? RNA is normally made from what? DNA. Okay? So in the jargon, normally cells have a DNA-dependent RNA polymerase. The DNA-dependent means it's using DNA as a template. These RNA viruses need an RNA-dependent RNA polymerase. So they're going to have a novel enzyme in order to replicate their genomes. Okay? Now, here's the key point. I said previously, and I always have to keep modifying things, that viruses were essentially just a piece of nucleic acid wrapped up in a protein coat. If you're a positive strand RNA, that means your RNA is in the right orientation to be used directly as a messenger RNA. So when that RNA enters the cell, it looks like a messenger RNA to the host cell. The ribosomes interact with it, and it begins making the viral RNAs, and one of them will be this RNA-dependent RNA polymerase. A negative strand RNA means that the RNA is the complement of the messenger RNA, and the first thing it's going to have to do is make an RNA from that negative strand RNA to make the positive strand. You with me? No? Okay, so the positive strand RNA is pretty straightforward. The RNA is functionally a messenger RNA, and the ribosomes are ready to go. A negative strand RNA is the complement, the reverse. Just like DNA has a complementary strands, right? RNA can do the same thing. The negative strand RNA is the complement of the positive strand. So the cell has to first make a positive copy off of the negative strand, then it can interact with the ribosomes. So a negative strand RNA is not a functional messenger RNA. So to even get this whole process started, it has to bring that enzyme with it. So the main thing is the negative strand viruses have to have packaged in that virion a functional copy of an RNA-dependent RNA polymerase, because it's not going to be there when they infect the host cell. The positive strand RNA virus doesn't have to do that because its RNA is already a functional messenger RNA, and the host cell will make the enzyme for it. Okay? So it starts to get us into thinking about the molecular structure of these molecules. All right. And the third one is the retroviruses with the reverse transcriptase. These viruses, the genome is a single-stranded RNA, but it gets reverse transcribed. That is, the RNA is used as a template to make a DNA molecule. So transcription is normally the DNA is the template, the RNA is the product. This is reverse transcription. RNA is the template, DNA as a product. It actually makes a double-stranded DNA, and then that double-stranded DNA inserts itself into the genome just like a provirus, just like the episomes, just like the bacteriophage we just looked at. So you can kind of see this evolutionary link, if you want to, between these processes that occur in bacteria are actually also occurring with these viruses infecting animal cells. In this case, we talk about HIV, we're talking about human cells. So these viral genomes, again, needed a unique enzyme. So they needed a RNA-dependent DNA polymerase. This was a reverse transcriptase. Oh, I think I got ahead of myself. Here we go. Okay. So, would you expect to find expect to find reverse transcriptase in your cells? No. Okay. Later on, when we talk about the enzyme complex called the telomerase, I think we've mentioned it already. I'll try to make an argument that it functionally is a reverse transcriptase, but except for that little special case, no. This is, again, these are viral specific. So again, the HIV virion has to carry a functional copy of the reverse transcriptase with it in order to be able to initiate the process, because if it infects the cells, there isn't going to be an enzyme that will make DNA from that RNA viral genome. Okay, and so it inserts itself as a provirus, and under the and again, this is one reason that HIV can be latent for such long periods of time, is that provirus can sit there in a quiescent form. The genes aren't really expressed, the viral particles aren't really being made, and under the appropriate conditions, then they start being expressed, and the virus, and you start getting viral particles. Another major difference between these lytic infections in bacteria and a lot of these animal viruses are in these bacterial infections, these lytic infections end up killing the cell. They degrade the host genome, they lyse the cell, and the cell is dead. With many of these animal viruses, so you're talking about these eukaryotic viruses, these viruses actually carry on a chronic infection. And so the viruses can actually bleb, is the term, they pass through the cell membrane. The cell membrane is what? A phospholipid. So think of it like a drop of oil. So you can have the viral particle can pass through it, and in many cases ends up being coated by the cell membrane material. That means the virus now carries many of the surface antigens that are on the normal cells, and not only does it not kill the cell, but also it helps evade the host immune system because they look more host-like in that sense. So eventually the cells do die, but in these animal infections, you tend to get these chronic infections where the viruses are just blebbing off, either from the nucleus, you can see them. I remember years ago we were working with some HeLa cells, which are a tumor cell line, and we were doing some EM micrographs on them, and you could just, they had been infected with some kind of virus, and you could just see these just hundreds of these viruses just blebbing off from the cells. So that's, yeah, basically that's what I was saying right there. All right, so just a little bit about HIV. You probably already know this. So the main thing is that it attacks the immune system. It took a long time to find the receptor for the HIV, but it's on the helper T cells. And basically it's an impairment, it manages, it's a very smart virus, it manages to impair the immune system. HIV patients end up being strongly immunocompromised, and usually it's some type of a secondary infection is what ends up killing them. I think it's pretty amazing in terms of what these viruses, in the context, only from a scientific, not from a humanitarian standpoint, is HIV has been studied since the 1980s. So there's been millions of dollars spent on it. We've sequenced it. We know every gene about it. We know every nucleotide about it. And yet still really haven't been able to bring the disease completely under control. The last slide is just a little evolutionary slide. that HIV most likely, it looks very much like one of the simian immunodeficiency viruses. So this is the sort of thing that you see, these evolutionary jumps, where the virus initially was only able to infect the monkeys. There's been some mutations, and then it's able to make a leap to a new host. In many cases, if these were often very remote areas of the world where there wasn't a lot of travel, worldwide travel, these diseases remain very confined. Now that we're such an interglobal community and so much transport, that's why things like Ebola and such, you're starting to see that being popping up. It's mainly because of the highly mobile nature of our world today. Okay. So any questions? So that's the chapter on bacterial. Yes.
Some cancers and other things that are caused by viruses, some people believe there are other things that are caused by viruses that we don't know. What would the genetic markers be? What kind of genetic markers would you look for, and could they be hidden for things like that?
Okay, I don't know if I know. So the question is, there are some viruses that have a very tight association with a few cancers. So probably back about the 1980s or so, when these oncogenes were discovered associated with certain viruses, it was like, this is the key to human cancer. They are viral in origin. But then it was realized, no, what was really happening is these viruses were acting as vectors and carrying normal human genes with them, except they were expressing them in the wrong way at the wrong time and such. So cancer basically is a genetic disease because it involves 2 or more genetic mutations. Viruses can cause those mutations. Other things can cause those mutations. Whether other diseases are associated with viruses, I wouldn't be surprised. The viruses can act as vectors for carrying genes that they picked up from one host cell to another.
And so when you're looking at someone's genes, what would you see that would show you viral interference?
If there was some unique sequence to the viruses, it would jump out. The problem is that a big part of our genome looks like defective retroviruses. And so a lot of the so-called junk DNA, which is now called the dark matter of the human genome, would look very viral-like, so it might be hard to tease that out. But the genetics is just getting so detailed that I think I'm giving you an incomplete explanation. Yeah, I mean, the tools for human genetics in the last decade have just exploded and just really quite amazing now. All right, anything else? This is all we're going to do on bacterial genetics, and I don't think we're going to do much with viral genetics. From here on, we really move into the molecular aspect of the course, which is really where genetics is today. So this is chapter 10. So this will be the last chapter for exam 2. Now, a lot of this, as we go through these different, DNA what is the genetic material? What's the evidence for it? DNA replication, DNA repair, transcription, translation, gene regulation, gene expression. I take it back. We do have one chapter where I'll tell you more about the lac operon than you ever wanted to know. You're going to see this in other courses. So you might go, well, sometimes it might be an advantage. I just happen to remember when they were doing the TCA cycle in probably organic chemistry, biochemistry, probably in biochemistry. It was also being covered in microbiology at the same time. So I got 2 different versions of it. So what we're going to be doing is looking at these processes more from the geneticist point of view, as a molecular geneticist, from a functional standpoint, and not so much the enzymology of it. So I'll leave it up to the biochemist to make you memorize all the enzymes that are involved in the structures. Do they still make you do that? Or have they wimped out now? You know the reason you have to memorize all those pathways is because I had to, okay? There's actually a real legitimate reason. But anyway, so what you're doing is don't try to separate and say, okay, this is something separate from biochemistry. They all come together. You're getting the same topic, but you're getting different points of view on it. And if you integrate all those together in your mind, it should give you a better understanding of what we're doing. So this chapter is on the DNA and the nature of the gene. That it's obvious now that genes are encoded in DNA. It wasn't always that way. So what I want to do is start the discussion by asking, okay, what are the requirements? What are going to be the requirements for the genetic material in a eukaryotic cell? And we're focused right now on well, I can't take that back. The examples I'm going to show you are actually from bacteria because that's what was available. We got really three requirements we got to look at. It has to encode some very complex information. Whatever the molecule is that's carrying the genetic information has got to be able to carry enough complex information to give all the information to put together a human being. It's also going to have to replicate itself with an extremely high level of fidelity. That is, it's got to be able to replicate itself and only make very few, very rare mistakes. And it somehow has to encode the phenotype. There has to be a relationship between what information I see in this genetic material and the phenotype of the individual. Okay. Okay. So we talked about Mendel's work, which was in the late 1800s, 1860s or so. We talked about the discovery of the chromosome theory of heredity. So a lot of the chromosome structure, meiosis, the discovery of the sex chromosomes and such, and that the genes, this genetic information must be associated with chromosomes, was probably also done in the late 1800s, the late 1800s. And this molecule we know as DNA, deoxyribonucleic acid, chemically was also known by the late 1800s. So by the beginning of the 1900s, all of the information was in place to really understand how the genetic system works, but it was going to take about another 50 years to actually put all those pieces together. So what was known as late as those 1800s, biochemically, and people were looking at eukaryotic cells, so they were looking at chromatin, because they're looking at it under a microscope. They knew that this chromatin, these chromosomes, was about 50% DNA and 50% protein. It was also known to contain these four nitrogenous bases, adenine, cytosine, guanine, thymine, which we just all know as A, C, G, and T. So biochemically, that part was known. But here was the problem. The scientist, Levine, spent 40 years studying the structure of DNA and got it wrong. Okay? So he was studying the structure of DNA. We knew about these nitrogenous bases. But what he incorrectly concluded was that A, T, G, and C were each present in equal molar amounts in DNA. Part of that was because of the particular DNA sources that he was using, and the other was a limitation of the technology. So from that, he concluded that DNA would have this very simple little tetranucleotide structure. So imagine it was just a circular molecule and had one copy of each base on it. Well, if you think about the chromatin, and you knew about proteins and all these different enzymes, looking at this, it was like, how could you possibly encode genetic information? And so the DNA was probably just some structural component of the chromosomes. So for a long time, what this really did was lead to the idea that the genetic information must be actually encoded in protein. And this idea persisted until the 1940s, until some experiments that we'll talk about just said, no, there's no other explanation other than the genetic material must be encoded in DNA. Okay, so before we go into that, let's try this question. It's going pretty fast, so we'll go 1 minute. Okay, 3, 2, 1. All right, the answer is B. I just made that one up. Okay. Any questions? All right. So Levine's idea persisted until Chargaff came along in the 1940s and 1950s. And again, as I've said, a lot of what we've learned, especially on the molecular side of genetics, really depends on new technology coming along. So Chargaff used this new technique called thin layer chromatography, which I don't know anyone who uses this anymore, but it was new about 60 years ago. And two things worked in his favor. One was his ability to quantitate the amount of each of the 4 bases in DNA was much more precise with this thin layer chromatography than Levine's. And the second is he took DNA samples from several different sources. In doing so, what he found was, you may already should be somewhat familiar with this, that if he takes a DNA sample, it didn't really matter what the source was, whether it was prokaryotic or eukaryotic, animal, plant, didn't matter. That if he looks at the molar amount, what do I mean by molar amounts? Just means there's equal moles in that particular sample. So that the amount of A was always equal to T, and the amount of G was always equal to C. But what he found that differed from Levine and was critical was the amount of G plus C varied depending on the source of the DNA. Now, I know G and C base pair with each other. I mean, if you're going along the DNA, a single strand, what percentage of it is G plus C residues? And so this can vary anywhere from maybe as low as 30% of the DNA, which means it's got lots of A's and T's on it, or it can be much higher. And it varies. And so what he really showed was you don't have equal molar amounts, but rather A was always equal to T, G was always equal to C, but this G plus C content could vary depending on the source of the DNA. This was a critical piece of information that Watson and Crick used when they put their structure together for the DNA molecule. Okay, so using Chargaff's rules, if you have a new DNA sample and it's 30% adenine, what percentage of the genome would be guanine? I'll go to 1 minute on this. It looks like people are getting this pretty fast. What? I do. Why do you miss them? I don't. Sometimes I've done that. I've taken clicker questions and just like, let's just see what happens. And you answer it in class, but you don't answer it on the exam. I don't know how that works. All right. So what's the answer? 20. Okay. So if it's 30% A, it's 30% T, that's 60%, that leaves 40%. Half of that has to be G, so 20%. Okay? All right. So here's a very cool experiment. This experiment goes way back into the 1920s, I think 1928, something like that. So this was the discovery of transformation. This is where the term transformation comes from. So we talked about transformation as a mechanism of getting DNA from outside the cell into the cells. And this is something we do routinely now for molecular cloning purposes and such. It goes all the way back to this experiment of almost 100 years ago done by Griffith. So he's infecting mice with some strains of Streptococcus pneumoniae, which the virulent forms are lethal in the mouse. So he'd isolated several different strains of the Streptococcus. Some are virulent, so they cause the disease and they kill the mouse. Also, when you grow them on a petri plate, on agar medium, they give you a nice smooth colony morphology. And they do that because they have a polysaccharide surrounding them that helps protect that bacterium from being detected by the immune cells. And so you get this smooth colony morphology. So these were called S strains. Type 3 was just the immunological strain of it. So we have type 3S is virulent. He also had some avirulent strains. That is, he could infect these into the mice, and then the mice were happy. There was no problem. There was no infection. They had a rough morphology because they lacked this polysaccharide coat. They had a mutation in the genes that encode that. And because of the lack of this polysaccharide coat, they were very effectively picked off by the mouse's immune system, and therefore these were avirulent bacteria. And they are immunological strain 2, so type 2R are not virulent. Okay? So, if you inject the mouse with type 3S, what's going to happen? The cell's going to, they're going to die. That was lethal. That was expected. Okay? You inject them with the type 2R, harmless. Now, if you take those virulent cells, the 3S, and you kill them by heating them, so if they're dead, you inject them into the mouse, and the mouse is fine. Does that seem like an expected result? Okay. Now, take those dead virulent cells that don't kill the mouse, mix them with the avirulent R cells, which don't kill the mouse, inject that combination, and you kill the mouse. Was that an expected result? No, that was the interesting thing, which made it a publishable result. Okay, so let's see. So here's the type 3S. These are virulent. You inject them in the mouse, and you get a dead mouse. The non-virulent, inject it in the mouse is fine, facing upright. You heat kill those virulent mice. I'm sorry, the virulent bacteria. No, I'm not going to do that to small animals. Okay, so you got dead virulent bacteria. The mouse is alive. Now you mix the dead virulent bacteria with the live avirulent bacteria, and the mouse is dead. Plus, when you do a necropsy, which is an autopsy on an animal, you get out virulent bacteria. So you had live avirulent bacteria, dead virulent bacteria, and somehow those avirulent bacteria were transformed into the virulent bacteria. He didn't know how that process worked. It was just a reproducible experiment. So that's how the term transformation came about. So a very interesting observation, but he really couldn't explain it. Yes?
So when you get the mixture ones and they come out virulent, that's not an example of what we just talked about with them being lysogenic jumping out of that lytic?
No, there's no viruses involved here. Right. So there was just some substance now in these heat-killed bacteria that were somehow genetically changing the harmless bacteria into deadly bacteria. Avery-MacLeod-McCarty worked on this problem for more than 10 years, trying to figure out what that transforming factor was. And what they showed was they could repeat this experiment, but in fact, they could narrow it down and show that it was in fact DNA was the transforming material. Here's how they did it. So they started out with the virulent bacteria. And what they mean by a filtrate here is, one is they homogenized the cells. They broke the cells open and just recovered the soluble portion of the cells. So one is they showed they could do this experiment. You didn't have to have an intact bacterium to do it. It was something soluble in the cells that was transforming. So you heat kill the bacteria, you homogenize them, filter it out. So you just really have the soluble portion from these heat-killed bacteria. Then you take one sample and treated it with RNase. So this would be an enzyme that would destroy the RNA. Another sample, you treated it with protease, an enzyme that would destroy the proteins. And another sample with DNase, an enzyme that would destroy the DNA. All right. Then you basically do the experiment by adding a sample of that filtrate to your type 2R. Those are the virulent or non-virulent? Those are the non-virulent. Okay. And so what they found was that you could inject this into the mice. Or actually, you didn't even have to. They found it just in the culture. Out of this sample, they got the virulent type 2, just like they did with the bacteria. They got it here, in here, but not here. These were the samples treated with protease and with RNase. So removing RNA and protein from the sample still gave you the transformation. It was only when you treated the sample with DNase that you lost the transforming activity. So what do you conclude from that? That the transforming activity that Griffith observed 10 years earlier was in fact DNA from the heat-killed cells were now being taken up by these avirulent cells. The genes required to make that polysaccharide coat were present on that DNA that was getting incorporated and genetically changing these avirulent cells into virulent cells. And this was a process called transformation. Okay? So it took them 10 years to do that experiment. We could do that experiment today. We could make it into a nice bioprinciples. Yeah, we could do it as a bioprinciples lab. It'd take an afternoon. Why can we do it in a couple hours and it took them 10 years? Because you're a lot smarter than they are, right? Okay? That's not the answer. Okay. Technology, that's always the answer, isn't it? And it is. We need 3 enzymes. We need RNase, DNase, and protease. How do I make those? No, it's not quite that difficult. I sit down, I get myself a cup of coffee, I sit down, and I go to the Promega website. If I place my order half an hour ago, FedEx would deliver the enzymes on dry ice tomorrow morning, and then we would do it. Yes, we can buy highly purified enzymes. When these investigators were doing these experiments, they had to make their own enzymes, preps. And I'm sure they had lots of DNA sample. Let's say you got a protease sample, but it's got a small amount of DNase contaminating it. So you do the experiment, and all of a sudden, wait a minute. Yesterday I treated it with protease, and I got transformation, and now I treated it with this new prep, and I didn't. So it was a matter of having to work out all the chemistry and isolating the enzymes and such. Now we purchase ones made by recombinant techniques that are highly purified, no cross-contamination. So yes, technology is the answer. Okay. Any questions? So let's try this question then. Well, lots of different opinions. Oh, we'll go to a minute and a half on this one. Yes.
Are you sure it's written?
I don't know. I guess we're going to find out. We'll go with my answer and then we'll discuss it. I mean, I've never done one of these questions and gotten them wrong before. So. I don't know. Let's see. All right, we'll go through it. We'll see what happens. Okay. What do you think the answer is? You don't... All right, let's see what happens here. Okay, so they took.
Just a protein.
Wait a minute. What the hell just? What do you mean just changed it?
It was protein. It was the voting machine.
Son of a bitch. I'm trying to find the other question. Well, the crazy thing is now I can't find the others. I can't find the original slide.
Sure you can.
We're just no. But from there. Oh wait a minute! I gotta go. Oh, it's right here. But okay, so I'll give you that one. Okay, so let's just see what the real thing's going on here. So this is a hypothetical question. If they had found that the heat-killed bacteria treated DNase and protease transformed the bacteria, but not treated with RNase, the conclusion would be RNA. You're saying the other slide said protein.
Instead of RNA on.
Okay. So does everybody understand why it would be RNA? I will give you that question. As soon as I find the slide, I can't even find the slide at the moment. Okay. So again, those are two of these landmark experiments, which were the main reason for going through them is to let you see the thought process. You take what you understood at the time, think about what possible outcomes you think are going on here, and then design an experiment to test that possibility. And they're not necessarily highly complicated experiments. Sometimes some of the best experiments are pretty simple in nature. Okay. The last couple of minutes, let me just introduce the other experiment, which is also a landmark experiment, the Hershey-Chase experiment, which was another approach to the same question. So again, at the time that this paper was published by Avery-MacLeod-McCarty, there was still a group of scientists who were still convinced that genetic material had to be encoded by protein. Scientists are supposed to be objective in their analysis of the data, but they're also humans. They're convinced that it was protein. So Hershey and Chase come along with another experiment that provided compelling proof that the genetic material had to be encoded in DNA. This was a bacteriophage. So they used a bacteriophage called T2, but its life cycle is very much almost identical to what you already saw with Lambda phage. All right. What they knew back in 1952 when they published this paper, when the paper was published, they knew that if they took purified bacteriophage T2, they had about equal amounts of DNA and protein. So there was no RNA there. It was just DNA and protein. One of those two had to be the genetic material. They also knew that the phage attached to the surface of the cell and that the phage progeny were coming from inside the infected cell. So whatever the genetic material was, it had to have been transferred into the cell. Okay? So the question is, how do we design an experiment to figure out whether it's either protein or DNA that's being transferred into the cell and possibly even found in the progeny particles, phage particles? So during most of this time and up until about 10 years ago, a lot of these experiments all used a lot of radioactivity. So we had to label these molecules in some way so we could distinguish them. So they grew one batch of E. coli infected with phage T2, but they put radioactive phosphorus in the medium, P32. So they know the phage are protein and DNA. Which molecule is going to get labeled by the phosphate? The DNA. It's not going to label the protein. In a separate batch, they did the same thing, but now they put S35 sulfur, a radioisotope of sulfur in there, which would label the protein, but not the DNA. So they had one batch of bacteriophage where the proteins were labeled with radioactivity, and another batch where just the DNA was labeled. Okay, then they isolate those bacteriophage, infect the cells, let the phage transfer whatever material is going to go into the cells, and then our old friend the Waring blender use that to generate shear forces to throw the phage off the surface and ask what molecule is inside the cell. Okay, so here's just an electron micrograph. So here are the bacteriophage attaching to the cell. So the whole question is, which molecule is getting transferred into the cell? So I'll just go through this, and then we can come back and review this when we come back on Wednesday. So here's the bacteriophage. It attaches to the E. coli. Something, in this case we know it's going to be DNA, gets transferred into the cells. As I said, it also makes an enzyme that starts breaking down the E. coli genome. So that's what these little fragments are. But as the phage particles get packaged up, the some of the radioactive DNA gets put into the phage particles, and you can isolate phage this way. Okay. Since everybody's getting restless, we'll do the analogous. We'll come back. We'll do this. So in one, we'll analyze it for the P32. The other, we'll analyze it for the S35. And from that, we'll be able to show that, in fact, it's the DNA, not the protein, that contains the genetic information. All right. So we'll call this a day. We'll see you again on Wednesday. Yes? Yeah, I think I did say 6 through 11 on one of them. Let me clarify. I'm 99% sure it's just 10.
Another question.
Yeah.
What we were just learning about how viruses don't really die.